Hello everyone, this is Chaihan. The paper early stage of biofilm assembly on microplastics is structured by substrate size and bacterial motility was published on iMeta by Ocean University of China. Microplastic particles are defined as plastic fragments with a diameter less 5 mm, and the surface of MPS promotes the settlement of bacteria and the formation of biofilms. The biofilm on MPS has environmental impacts such as influencing element cycling in marine ecosystems and causing species invasion. Bacterial settlement on MPS surface is affected by factors such as polymer types, exposure time, seawater salinity plus color, and some studies have focused on the relationship between MPS size and the functional characteristics of biofilm, such as in wastewater treatment systems. So we propose a scientific question. Does the size of MPS affect the assembly of in-situ biofilm communities in the ocean, and what are the specific mechanisms involved? Place the two sides 3 and 0.3 mm of microplastics and glass particles in the intercept zone and recover the developed biofilm after 10 and 20 days. At the community level, metagenomics, citing as RNA gene applicant sequencing, metatranscriptomics, and qRT-PCR techniques. At the fungal bacterium level, physiological and biochemical experiments including bacterial motility assays, MPS colonization assays, bacterial co-culture experiments, and genetic knockout experiment validation. And we found out the community structure of 0.3 MP10 is relatively unique, with the highest abundance of gamma-protein bacteria as the main dominant genus. And in PCOA, there is a significant difference between the 0.3 MP10 and other biofilm communities. And then the one we unknown one decided to read out to us almost all motility-related genes showed significant differential expression in the enrichment in 0.3 MP10, and the flazy flag-E genes are mainly present in the biofilm communities of automonas and some members of the respect group. The relative expression levels of motility-related genes in the 0.3 MPS10 biofilm community are higher than those in the 3 MPS10 community, such as flag-F genes. qRT-PCR analysis showed that the expression level of flag-E in the 0.3 MPS biofilm community was significantly higher than that in the 3 MPS community, and it was only expressed in the early stage, 5 or 10 days. Bacterial strain belonging to the automonas named PM193 was selected to test our discovery. The motility experiment showed that it has strong swimming ability. The MPS settlement experiment showed that the settlement of PM193 on 0.3 mm microplastics was related to bacterial movement. The other genes selected showed similar results to PM193. And the co-culture settlement experiments of motile and non-motile bacteria showed that the motility enhanced the settlement ability of bacteria on 0.3 microplastics. And the vibrio-immunity strain with knocked out flagellar genes significantly reduced its settlement on 0.3 microplastics. We summarized the following points. The community structure of the biofilm on the surface of 0.3 mm microplastics is significantly different from that of all other biofilm samples. Compared to 3 mm microplastics, the biofilm on the surface of 0.3 mm microplastics is enriched with genes involved in flagellar motility and chemotaxis, indicating that the community is more motile. Laboratory experiments with isolated bacterial strains confirm a strong correlation between bacterial motility and their colonization on 0.3 mm microplastics. Again, we thank iMeta very much.